Hello all. First of all, thank you, Protochips, for this opportunity to present some of our work. So this work was done at electron microscopy lab at Institute of Nanotechnology, which is headed by Professor Christian Kubel. So we worked on some high temperature dynamics of nanocrystalline graphene, and we did some time resolved high resolution imaging at high temperatures using Protochips fusion system. So before going to the results, a brief introduction about nanocrystalline graphene. So nanocrystalline graphene is formed by the thermochemical decomposition of thin polymer film. So you vacuum anneal a thin polymer film. This is a catalyst-free process which inhibits the growth of the domains, and the domain size or the crystallite size fall into order a few nanometers, which is why it is called a nanocrystalline graphene. This is a highly dynamic process with a number of intermediate reactions that is happening in between. So you have a initial polymer structure where you have the possibility to control the size, shape, and thickness of the material. And more importantly, you have the possibility to control the properties of the material by controlling the crystallite size, that is by controlling the graphitization temperature. So this unique properties made this an interesting candidate in a number of different uh, technological applications. So when we think about a property specific material, to realize a property specific material, what we need is to have a better understanding of the graphitization and the growth that is happening. So that's why we started thinking of doing some in situ transmission electron microscopy. And that's where we used the in situ heating holder from the protochips, where it has a MEMS based chip, contains freestanding membrane which act as both the sample support and the heating element, which provides a very confined heating. You have a very flexible heating rates, and more importantly, these chips are really stable at high temperature, which enables you to get some high resolution images at high temperatures. So we used both the Eduro and the fusion holder from the protochips, and with the different set of chips that they have with different coatings over the heating membrane. We use the chips that contains no coating over the holes, and as I told, it provides you with very high stability at high temperatures and very flexible heating rate that we can use. So to make the sample for this in situ graphitization, what we did was we took this chip and we spin coated it with a graphitizing polymer, and we did initial heating outside the TEM under similar vacuum conditions until 600 degrees Celsius. So this is basically because of a large amount of gas evolved during the initial pyrolysis process. So after this initial carbonization, what we observe is that there is a large area freestanding partially carbonized film formed across the holes of this MEMS based heating chip. And from Raman, we can see that the graphitization is similar to the substrate supported graphitization. So what we have here is as I told, a large area freestanding thin film, which is really ideal for microscopy. And we heated this inside the microscope from 600 to 1200 degrees Celsius. And we imaged at every temperature. We took some high resolution images at every temperature to see how the microstructure is evolving. And also we did some diffraction analysis to understand the crystallite size evolution, and also yields. By comparing it with a completely graphitic sample, we could calculate the sp2 fraction. And as you can see, the sp2 fraction increases and saturates at 1000 degrees Celsius. This leaves us with a two-stage growth mechanism. The sample is almost completely graphitic at 1000 degrees Celsius. So the intermediate temperature growth is mainly by consuming the disordered carbon around the crystallites, and the high temperature growth is by the merging of the already formed crystallites. So When you heat the sample to 1200 degrees Celsius and do some high resolution imaging, you can look into the structure of this. So you can see that the structure is highly defective with lot of defects and disordered edges. And there is also a lot of carbon nanostructures which is distributed across the structure. These carbon nanostructures are highly dynamic at high temperature. These small nanostructures are migrating and merging at high temperatures. So this dynamics is really fast. And to understand more about this high temperature dynamics of this merging, migration and merging of domains, we did some high resolution imaging, time resolved high resolution imaging at high temperature. So that's what we will see in the next couple of slides. So here, this is a 12 minutes dynamics at 1200 degrees Celsius. You have a small flake that is indicated by the blue arrow, which is rearranging and merging with the flake down, facilitating vertical material transfer. And at the same time, you have a flake indicated by the red arrow, which is jumping and attaching to the flake edge nearby, facilitating a lateral material transfer. So with this time resolved high resolution imaging at this temperature, we could identify migration and merging of these flakes as a mechanism that is active during high temperatures. Not only this, when these small flakes are migrating, as you can see, a small flake will migrate into the red circle and it will pin to a defect there. It is pinned there. So these small structures, while it is migrating and pinning, it is shrinking in size. And at the same time, you have some big flakes indicated by the blue arrow, which is growing by the addition of atoms. So this means that atoms or group of atoms can detach from the small flake and attach to the larger ones, facilitating something like an Oswald-like ripening in this highly defective systems, uh, which was not reported previously. So here we can identify two different mechanisms. One is the migration and merging, and where we have both lateral and vertical material transfer, and also you have the Oswald-like ripening by the movement of atoms or group of atoms. Now, one of the obvious questions that comes with these in situ measurements is that what the beam is doing here, or how can we separate out the beam-induced transformations and the inherent temperature-driven mechanisms? So for that, we did some beam off heating. So we uh, imaged an area and switch off the beam and image the same area after a particular time. And as you can see, all these dynamics that we observe during the beam on condition was also observed during the beam off condition, and also shows the stability of this heating heat MEMS based heating chip from protochips. You can see it's easy to find the same area after this time of continuous heating. So all these images, that all these dynamics that we observed during beam on condition was also there during beam of heating, which confirms that these are inherent temperature driven mechanism and not beam induced. And also you can see that when the beam is on, the dynamics is largely accelerated, which is obvious that the beam is providing some extra activation energy, which is accelerating these dynamics that we are observing. So to conclude, we could look into the high temperature dynamics of NCG using the fusion holder from protochips. We identified migration and merging as a prominent growth mechanism at these high temperatures, where you have both lateral material transfer as well as a vertical material transfer happening. And also we could see Oswald-like ripening in these material at high temperatures as another growth mechanism. And we also did some MD simulations to look into some of the activation energy associated with this. And then what we learned is that the activation energy for atom removal from the small flake comes down drastically in near defects. So that the flake can lose atom to a defect in the substrate, and it can also migrate through the active edges and extended line defects. In that way, an extended defect can act as a pathway between the shrinking flake and the growing one. So this Oswald light ripening is more defect mediated, uh, and we confirm that these are inherent temperature driven mechanism and not beam induced. But the beam is obviously giving some extra activation energy, thereby changing the reaction dynamics. So that is from my side, and I thank you all